This morning, I'm really excited to introduce Dr. Thomas House, who is a professor of mathematical sciences in the Department of Mathematics at the University of Manchester. He's a fellow of the Alan Turing Institute and is also a Royal Society Industry Fellow with IBM Research. And his research, I would say, I guess currently, but also prior to COVID, really focused on mathematical epidemiology, and some of his interests included efficient statistical inference using network data and model simplification for epidemiological models. During the COVID-19 pandemic, he's been assisting the UK government and public health officials as an expert on the Scientific Advisory Group for Emergencies, or as we know it, SAGE. And he's also an investigator with the Juniper organization, which we heard a bit about. It's a collaborative effort across many universities in the UK. And we heard a bit about it from Julia Gog a few months ago. So we're really excited to have him here to discuss work using household infection surveys to learn about COVID-19 transmission dynamics. So I'll hand it over to you.
Oh, thanks very much. That's a lovely introduction. So I'm going to talk about this particular study we've had. So, you know, the UK hasn't had a sort of far from perfect response to the pandemic, but I guess which government has got everything right. But one of the things that was done right was, I think, the scientific response has an awful lot to be proud of. Some of you might have heard of the recovery trials showing the efficacy of dexamethasone, and another thing that has been very good is well, there was this set of things funded. This is this slide here called the National Core Studies. So rather than make certain very key efforts have to bid for funding competitively, things that were providing a kind of core service were funded under this. And things like the recovery trial already had what they call sleeper funding kind of ready to go as soon as there was a pandemic. And a lot of this credit, I think, goes to our kind of equivalent of Fauci or whoever, Chris Whitty, who's the CMO, who's very, very sensible, has taken an enormous amount of stick. You may not have noticed, but got in trouble with Nicki Minaj on Twitter at one point, and he's you know, this is a fun, a kind of amusing clash of worlds if you want to look up that story. But one thing he got really deserves a lot of credit for is getting this funded. Him and the chief scientific advisor Patrick Vallance were largely empowered to get on with their jobs and got a lot of this stuff ready. On the issue of masking, one of the things that was funded was the PROTECT study, which Kath Noakes is quite prominent in looking at environmental transmission. But I've kind of set myself a rule of trying to avoid talking too much about that because it's best left to specialists like that. But looking for the best evidence about masking in types and so on was part of that. Anyway, but the thing I have been involved in is this community surveillance. So the REACT study was run out of Imperial College London, and this had an individual-based cross-sectional design. So REACT sampled a lot of people, but not continually. So it had waves, and in each wave they would get a lot of individuals sampled, but they wouldn't cluster within the household, right? So REACT was designed to get a sort of snapshot, and for that snapshot to be quite sort of geographically informative, even at low prevalences. I've been more involved with the Office for National Statistics study, which I'm going to talk about. And this was a household-based longitudinal design. So it recruited households and at the height up to about 500,000 people in kind of about 250,000 households. So quite a large percentage of the population for a study was recruited to this. So most people know someone who's participating in this study, and they're followed up. So this is much more, in some ways, this is much more informative data. It's sort of smaller in each sample than a REACT was, but there's also this extreme wealth of following people up over time and having the household study. And it turned out when this was being talked about, I was the only person or one of three people who'd written a study on design of household longitudinal studies. So I ended up in a room with all the great and the good of British science, like Jeremy Farrar and people, feeling rather out of my depth. But this is the basis on which I was involved. This thing that I thought was an extremely niche research interest, and I mentioned, I think in the abstract I submitted, the sort of Kenyan group had tried to do something like this, suddenly became one of the biggest studies, and I had to kind of put my money where my mouth was in terms of actually getting some results out of this data. There's also other data that we have is lots of case finding data. That's all been made public. Data on death and hospitalization. Working for SAGE, you get slightly higher resolution data, but an awful lot of this has been made public. And there's been two app-based systems, the COVID symptom study or Zoe and the official NHS app. A lot of this funding is now drying up. I try to avoid getting too political, but it's clear that a certain amount of the reason our government's doing this is that they were found partying and breaking the rules really badly during the height of lockdown, and are now trying to say, well, it's all right because it's all over. I don't think that's a terribly controversial opinion. They're not really hiding it. But science is unfortunate. This very good science has unfortunately been a bit of a victim of that real push to say that, well, everything's done and we don't need to worry about this disease anymore. But the ONS study has continued, which is good. And I think other countries it's something that other countries or maybe states in the US may want to think about as routine testing is reduced. If there's a surveillance aspect and they're persuadable that this is still worth following, a kind of community-based surveillance design is certainly some other European countries are picking up. Anyway, so that's the broader kind of political context and very slight bit of thing. And so the Office for National Statistics is an independent, or is a kind of is funded by the government, but is set up so that the government is not supposed to influence it, right? So it's accountable to a statistical regulator. And my experience of working with it is it works unbelievably hard to ensure the kind of fidelity and trustworthiness of any number it puts out, which is a bit of a pain because we kind of do that as academics, but we sort of equally, you always know that a paper might be found wrong, right? Whereas the ONS really has this thing where they don't like their numbers later being found to be wrong, which is something I found difficult, right? That there's a question, right? That in biology, it's very hard to get the number right very quickly, right? So their normal job is to say how many factories are there in this bit of the country, how many factories are there in Wales or something, where there's just an answer. But there isn't really an answer to what's the prevalence of coronavirus in Scotland, right? Or rather, you'd expect that number to get revised. So they've struggled a bit with that, but it's largely been recognised. They put out, you can find it. This is open to the world. You can download a lot of the charts from this site I've linked to here. I think it's a very they're a very admirable organisation that I'd not had direct contact with before. Okay, so why did it choose to do households? Well, one of the reasons is that people live in households, so our census naturally goes. The ONS is what runs the census. People live in households and they naturally sample households. But the other thing is that you can use households to infer something about transmission, right? So infections are special because you give them to each other. And so in the very early days of epidemiology, people used to kind of think, well, we need to make this into a proper science, so let's infect a load of mice. And it was actually in Manchester, where I am now, there was sort of probably millions of mice killed as people directly infected them and left them in cages and tried to work out how infection dynamics worked. And this wasn't terribly useful because we're not very much like mice. But I think what other people realised it. Oh, sorry, right. I've had to come into the office and it's on a motion sensor light, so I might do that another couple of times. Is that the household is like an ethical version of that experiment, the mice in the cage. So there are millions of households or thousands of households in your sample, and each one of them is a little transmission experiment, right? But it's one that would have happened anyway. So if we observe that, we're able to kind of have lots of replicate epidemics that we can learn things from, but that aren't, we've not deliberately caused them. That's part of how the epidemic was proceeding anyway. so that's my intuition, partly for why households are an interesting thing to look at. And this goes back to Reed and Frost, I guess, in the Baltimore epidemic and the Tecumseh flu study. So Irolongenium. Oh no, I'll talk a bit about that in the next slide, actually. Sorry, so before all the testing and tracing wound down, this coronavirus infection study tended to be like confirming what test and trace found, right? It was slightly frustrating in that sense. But now it's the main thing. So we're being looked at a lot more for like, well, now testing and tracing is stopping. What can this community surveillance tell us? Okay. So this is what I was starting to say. You get this close repeated nature of contact within households, and they're relatively stable. And the other thing is that interventions are designed at the household level. So generally speaking, people were asked when they were told to shelter in place or lockdown or quarantine or whatever, the local word was for it, they were asked to stay with their household members, right? So the household was kind of, and they'd often get things distributed to them on a household basis, right? So I've written here school LFD testing. That LFDs are what we call our rapid antigen tests, or I don't know, different countries seem to have come up with a different. So one of the things, the other things the UK got very right, and I think Mike Mina has been a very good advocate for these, is these rapid tests as a kind of, they were made freely available. And I think this was just brilliant. It's a very empowering intervention because it lets you know your disease status with a reasonable level of accuracy. So there was, everyone in high schools was encouraged to test twice weekly. But if you shared a household with someone who was in school, when the schools were open and not much else was, households containing school students were also testing, right? So there was a household element to the routine testing, as to the intervention-based testing, as well as this kind of surveillance testing. So quick bit of the history that I started on before, then I will get on to the equations. So Reed and Frost did this really interesting study in Baltimore. And the story was they simulated the first stochastic epidemic model pre-computers on a modified roulette table. Then Bailey did a bit, Bailey, I guess, came up with spectral methods in stochastic processes to analyze these sort of data. And I guess you had the Hope-Simpson studies of kind of spread of measles and things amongst children. So this was the early development of households, and it was always, so it's been, this goes back at least as far as the Kermack-McKendrick type equations, right? So the household type equations are slightly different from Kermack-McKendrick type population ones, but they have a kind of similar pedigree. Then later in the 20th century, Frank Ball derived a very general formula that generalized Bailey's results. Then Cheryl Addy, Iron Geelan, Betts Harrelaren did this really nice work on the Tecumseh flu study. So I think Tecumseh's in, is it Michigan? I don't know. But it was this very famous community study of influenza. And so that was analyzed using this final size formula, but people didn't have modern molecular methods. So as I remember, I think they had to try and grow the flu virus on pig's blood, which was obviously slower and much costlier. So now in the 21st century, the cutting edge of analyzing household data is actually, like for everything else, MCMC. And we have modern molecular techniques like PCR that make the confirmation of the organism involved much faster. But the issue is that so what I've been doing is using 20th century mathematics on the 21st century data because people want a quick answer. So I didn't have time to write an MCMC sampler that would work. So here's the study design. If you think about it, you've got time going along and you sample the households, and the household's got a stable composition of 4 people. And on visit 1 here, no one tests positive. On visit 2, person 1 and 2 test positive. On visit 3, person 2 and 3 test positive. And on visit 4, no one's positive, right? So this is what I mean by household longitudinal design. And this is, suppose this arose from this kind of tree, right? So a perfect test and trace system would give you this tree. It would give you the actual tree of infections because people would report as soon as they get ill, right? And the issue, and so what the UK well, what was Public Health England is now the UK Health Security Agency, did is they assumed that when they looked at household data, they assumed that the first person they found was the index case, which isn't always true, but it isn't a terrible assumption. And that all the other ones were secondary cases, and their main method of analysis was to do logistic regression on the secondary cases, right? Or some kind of binary regression on the secondary cases. Now, that isn't quite right, because some of the secondary cases will really be tertiary cases, and they might not have ascertained the first case correctly and all these other things. But in practice, it was fine, right? In practice, that might annoy us as modelers, but from a statistical point of view, it's fine. Particularly when they needed a quick answer about is this particular variant more infectious. But like I say, this very rich data is now going. So the first thing I'm going to talk about is just a very simple idea, which is, suppose I want to compare these two types of study, right? So in this one, they've assumed they know who the secondary cases are, and I don't know who the secondary cases are. But if you think about it, if I take a load of households that are the same type in some sense, so I group the households by household type, then I can work out what the secondary attack rate is in that group by type, because for each household with more than one case, I just take off one case and then divide that by the total number of people who could have been secondary cases, because I don't have to differentiate individuals within the household, okay? So the secondary attack rate is with a proportion of people who could have ever been a secondary case in a household, yeah? So it's this bit here is saying the households are size n, so the set of, I is the set of households that we're thinking about, yeah? So it's the set of households with at least one person infectious, so we don't get negative numbers here. And of those, we take all the people who could have been a secondary case, and that's the denominator, and the numerator is all the secondary cases, right? And that gives you a proportion or a percentage, right? That's the secondary attack rate. Now, so the idea is there are three, we split the data into three periods, one where alpha was dominant, one where delta was dominant, and one where omicron was dominant, and then split the households by group into ones that were not at all vaccinated, ones that were substantially vaccinated, and ones that were sort of intermediate. And you'll see the intermediate class bounces all over the place. So that was mainly done just so we could ignore it. And what you see is the secondary attack rate is much higher, even when adjusting for strain in the households that are unvaccinated, right? And it's the same for, and that's the same for each strain. So fortunately, the vaccines work. For transmission, this is separate to whether the vaccines work for susceptibility to some extent, and it's a total effect size. But it's indicative that the vaccine is working to stop transmission. And then we look at what the secondary attack rates are by strain at given levels of vaccination, and see that basically Delta and Omicron are more infectious within the households that are unvaccinated, yeah. But even for the vaccinated ones, you see there isn't really any difference between Delta and Alpha, yeah. Once you're vaccinated, the transmission, but Omicron is a bit more of a vaccine escape for transmission, yeah. So that this is kind of confirmatory on top of what we know, but it's nice to see. And then this is the kind of table.
Thomas, can I ask a quick question?
Yeah.
So on that slide, it sounds like you're controlling for infectious status, like, you know, having a documented infection versus not in the part D.
So D is all these households are ones that have got at least one infection in them, and during the period when a particular variant was dominant.
I see, but they're not vaccinated households. They're unvaccinated.
These are ones that were unvaccinated at the time the disease. So because it's been running since like months before there was any vaccine. So this has been running since April 2020, and we got Alpha because it emerged in the UK. We got it. It was pretty much dominant since mid-November 2020. So there was quite a lot of the Alpha spread. They couldn't have been vaccinated. So that's what I said. Positive first means if it was a household that had a positive before they could even have been vaccinated, So a lot of these households will have gone on to be fully vaccinated.
Right. So it's not controlling for if an infection had happened like at a previous time in the pandemic.
No. So there's prior immunity, I think, is not in this, and probably should. But the point is that at some point you can't do this stratifying the household into types and contain and maintain statistical power. So I guess that's why we called it total effect analysis, right? It's just a total effect, and there's only so much you can control for. So there's an awful lot of stuff not controlled for, but it's just saying there's a total effect in these households categorized certain ways. You can kind of see effects that we'd like to see, right, and we'd expect to see. So it's kind of confirmatory.
Okay, thank you.
And then these are the tables. So I'm, on my to-do list after this talk is to start writing this up, because the early preprint contained almost none of this. It was sort of rushed out, and now we've got a lot more to say.
Thomas, okay, would you please just redefine what you mean by vaccinated, intermediate, vaccinated first? What were those?
Yes, so that's because we had to group the household at the household level. So positive first is there was no vaccine in the household at the time of the first infection. Vaccine first is that there was at least one person in the household who was more than 21 days post-vaccine at the time of infection. And the reason for that is that means they'll almost certain the other adults, that means that it's almost certain that the other adults in the household will also be vaccinated. But because children are vaccinated much later, that you wouldn't find anywhere the whole household was vaccinated. But this is what I mean. So in the big bit of scientific work we have to do is what I'll go on to is designing our regression frame. We need a regression framework that includes vaccine more.
Okay, and sorry, and an intermediate is somewhere in between.
Just neither of those holds.
Okay.
So intermediate just means that someone is
recently vaccinated.
There was someone who was someone might have had their jab, and then like a week later the infection was in or something.
Thank you.
Okay, so this is the paper, the peer-reviewed paper with this. It was methods Tim Kinney, Julian and I wrote just pre-pandemic, and it's based on these kind of what I call the late 20th century ball ADDIE type final size equations, and all the code's open. I've like a lot of people finally moved from MATLAB to Python, so it's all open in Python for people who want to download and use this method. But the idea was that it's very hard to do this in real time, and what makes households hard is that your disease status is not conditionally independent on other people's disease status. So normally when you have a group, so suppose you have a household, you say, well, what a statistician would normally tell you is, yeah, we model household effect because I put a random effect or fixed effect or something on each household. And what that would be telling you is something like, the household will smoke, right? Or they have other unhealthy things that we've not captured by asking. If they smoke, they have all sorts of other unhealthy things they're probably doing, so we want to capture that correlation by having a household level effect. And the point is that but your outcome in terms of cancer or something is still conditionally independent on knowing that the value of that random effect and all your other things, like whether you smoke and how you eat and so on. But the infection is actually dependent on the outcomes that other people experience. So normally in artificial intelligence or stats or whatever, they call it the X's and the Y's. So the X's are the things you're trying to use to make the prediction, and the Y is the thing you're trying to predict. But my Y is not independent of your Y if we share a household. Yeah, and that's what makes household analysis squirrely, basically. And why you need these fancy equations, right? So doing that in real time is possible with MCMC, but at the volumes of data we have, it's just not fast enough to give people an answer in a useful time frame. And as I've said, we were slow enough already compared to people working with case data and logistic regression. So what I did is split things up into periods of time where it was very unlikely there'd be any reinfections, which I call tranches, we call tranches, and then use time as what statisticians would call an instrumental variable, right? So lots of things have changed with time. And so, but time is, we're going to monitor the same set of effects on household transmission and risk over time, and then try and interpret them in terms of things that have changed with time. So this is the first 6 I've done because that was when we got the paper through peer review, but we basically release a tranche every few weeks now. And that gets posted on the GitHub site that I link to, and we sometimes update the analysis a bit. So the variables that we start, but the issue is we start off here before the vaccine, so we didn't build the model to run with vaccines. So the thing we're currently working frantically on, and I think the issue is we all just hit burnout, and that's why it's been a bit slow. Anyway, so the reason the vaccination isn't here is we've been doing this since April 2020, basically. So what we were interested in is swab positivity, household size, the Alpha and Delta variant. This was in the first 6 tranches, age of participant, and work in patient-facing roles. I'll tell you a funny story as well quickly about work in patient-facing roles, which is at the very start of this, I was at a kind of, it was early enough on in the pandemic that I was in a Skype for business meeting with people at the ONS. And they said, oh, there's a real problem because we're going to ask people their occupation, but normally we do the census and it's going to take us a few months to code all the occupations. And they were worrying about this. And I said, but we kind of know that working in hospitals and care homes is worse. So why don't you just have one tick box on the front saying, do you work in a hospital or do you work in a care home? And they sort of ummed and did this. And then in the first analysis, it was the only thing that came out as statistically significant. And one of the applied statisticians said to me, Thomas, that tick box is going to be the most useful thing you ever do. And we're not into malice, it's probably true. All this technical stuff is probably less important than the fact we worked, because this was quite useful in advocating for extra protections for healthcare workers and people in care homes. The fact we could see just so obviously that they were at this elevated risk. Anyway, so the work in patient-facing roles was a tick box that I as a complete novice just said, oh, why don't you put this down? Right, so that was actually my most useful contribution to the COVID pandemic, was suggesting a tick box. So this is the size of them, and this is what's amazing, that I really felt, like I said, I felt a bit bad about using these slightly old-fashioned methods, and even presenting to people like Simon Koshmers or whatever. I was like, look, could you do anything else with these numbers? And the answer is no. It's just so big. The data is just so big. It's even in the first one, there was like hundreds of thousands, nearly 100,000 participants. There weren't very many positives, right? There's only a few hundred positives because it was launched when our kind of first lockdown had really bitten. Over the UK now, we're running at over we were running at close to 10% prevalence recently in this study, which is when this kind of thing is most useful, because actually at the same point that case finding breaks down because there are too many cases, random community surveillance is the best. So actually, I was, if other countries are thinking about it, I suspect lots of them are running at close to 5 to 10% prevalence of BA2. But we thought it was really big when things like Alpha and Delta were getting up to 1% prevalence, because when we started, it was this tiny prevalence, and we were thinking, oh, is it, are we going to be able to keep this down low enough that we never really need surveillance? And then unfortunately have this incredibly lethal second wave. Right, so just to be clear, this OR plus N plus S, do people know about this S positivity, S gene target failure as a marker of? I don't tell people things they know. So basically the order that the variants came in the UK, sorry, we went from having these 3 gene positives in PCR to 2 gene positives because of Alpha, then back to 3 genes because of Delta. And that meant that we could use PCR positivity as a reasonably good proxy for strain, whether things are Alpha or Delta. Okay, so these are the sizes of the things. So what's a household? It's a partition of the population into disjoint sets of integers, right? That's all I'm saying here. Just because I've given this talk at SIAM and places where they like you to be precise. What I'm saying is everyone's in a household and the households don't overlap, right? And there's a finite number of them. So before jumping into modeling, as I say, so we should do some exploratory analysis. So ZKL, which I've defined to with indicator functions. And so the other thing to say quickly is, although there are some households bigger than 6, they're very atypical. So well under 1% of people live in households of size greater than 6, and they tend to be odd in some way, in the sense of like that a household of size 6 is almost certainly one of a small number of configurations, like 2 parents and 4 kids, or but the ones of size over 6 tend to be more heterogeneous in terms of how they're composed. So we just stuck with the households of size 2 to 6, and a household size 1 has no transmission in it, so we don't really need, we keep them in for some things, but they're less interesting. So you see there are the 1 participants here. These histograms are telling you what is the count of positives in the household during the tranche, right? So during the tranche, we take the end of that and we say, right, looking back over this 2 months or 3 months or 1 month when there's high prevalence, so we get power quickly, how much of the household was infected. And because we follow them up, we can follow them up a few weeks after the tranche when we do the final analysis of it and look back and go, so we attribute each household to the first infection, that to the tranche where its first infection came in. Then if there are subsequent ones that take us outside that time period, they're allocated to the main tranche, right? And where the information is in this data is that these histograms are over dispersed compared to a Poisson or compared to a binomial. So you see for some of them, they take this U shape, right? Like this one here, it actually goes down and up, right? Which it couldn't do if it was a Poisson or a binomial. And although you can't see it because the ones with 0 would be enormous, right? Because prevalence is like 1%ish at most in these tranches. The amount that are 2 is much bigger than the p squared. Like if you wrote p for the proportion of infections, right? The number that have got 2 infections in is much bigger than p squared, right? So there's over dispersal in the counts, and that's where the information on transmission comes from. Yep, so this insight goes back to Bull, Mollison, and Scalia-Tomba in the late 90s. They plotted a load of these type things and said, look, this gives you information on how much transmission is going on between the households and how much within. There's loads of within households, you'd see something even more severe than this kind of U, right? You'd see a more severe U, and if there was almost none, it would just be binomial, right, within each household. But these are very, very fat-tailed compared to binomial. Although it's not obvious from this because, you don't see how big the 0 bar is. And the ideas about how to visualize that, maybe a log scale, I don't know. Right. So then I did this density plot. So there's this question of, I think this was the most kind of painful thing I was involved in the whole pandemic, really, because there's this question of, do kids get ill and do they transmit? And this has always been a problem because kind of obviously being off school is very bad for children, but we don't want them catching COVID and bringing it back home. So I think there was a lot of, it would have been rather convenient if we'd been able to conclude that children don't transmit very much. And we didn't see very many cases early on. So a lot of people were hoping that we could say, oh, actually children are almost a dead end for transmission. And there was talk about, they're ACE2 inhibitors are less or whatever. But it was just very unfortunate. So these plots are like, they're density plots of proportion of the household with under 16 positives and proportion of the household with over 16 positives. And so because you see some households that only have child positives, and this holds even when both children aren't in school, you kind of know that the children are bringing it back and transmitting. And we'll go on to the more formal analysis of this, but this was exploratory analysis just in case people get worried that the formal analysis is wrong. So this is kind of annoying because it means that then you have to say, well, we want children to be in school, but we also don't want, and it's just hard, right? I mean, I'm glad I didn't have to make any of the policy calls there, is all I will say. But the results are what they are. I think we'd hoped that the result would have been no or very little transmission amongst children. Right. So yes, these are these sort of chessboard plots I think people struggle with, but I think they also make clearer how the bimodality works, that you're not seeing a kind of concentration of middling proportions, but you see the action is on the edges, right? So I think they make the over dispersal clearer, right? That these are multimodal. Fundamentally, we've got a multimodal distribution, right underneath. It's not something that's going to have a nice central limit. So I also did a bit of we also did a bit of exploratory analysis. I'm saying I because a lot of these I did and got it like vaguely wrong until other people had had a go at checking. So I don't want to include. I want to give them the credit for helping me tidy some of this up, but a lot of it was just, you know, stuff I was just fiddling around and initially wasn't brilliant. But now I think this is actually quite a powerful way of looking at things, which is you count up all the pairs of individuals in households that have certain properties and then compare that to a null in which those properties marginals are randomly distributed within the household. And you look at the difference between the null and the null hypothesis of no association within households and how they're actually correlated. And you see this kind of, so this was mainly to look at, does it look like the variants are spreading? And so the fact you get these green squares, which are kind of positive residuals by gene pattern, is sort of indicative of the fact that the variants are more clustered in households than you'd expect from their community prevalence, which is again a kind of signal of transmission, right? So these are all these are all a bit fiddly, but they're ways of just making sure that the model isn't this sort of exploratory-ish analysis to make sure the model isn't overdoing things, right? So very quickly, the ball type equations come from this thing called the Selke construction. And basically each individual, you pick how long they're going to be infected for, then you pick an exponential 1 threshold for the time at which they're going to, the cumulative force of infection at which they're going to become infectious, right? This equation here tells you why that's kind of equivalent, right? That the probability that this random variable exceeds the cumulative force of infection at a certain time given it didn't before. Hang on, I've just spotted the typo there. I hope that isn't in the paper, or maybe it's the right one. Anyway, ignore that bit, but it's this. Oh no, hang on, no, that's right. Sorry, it's the probability that you remain. No, that isn't a typo. Sorry. The probability that you remain susceptible, right, is this thing, which is the same as the stochastic process, standard stochastic process definition, right? So that is the right thing. Okay. And then we try and write down some final size equations on the basis of that Selke construction. So if we can embed every household configuration of final size outcomes in the integers, which we can because they're just finite. So the map iota is the mapping of all household states on some set of integers. We can write the final size equations as a matrix equation. And then that matrix equation is easy to solve with linear algebra, right? So this resolves the problem I was talking about your outcomes depending on someone else's outcomes. This P here is a vector of the probabilities of every different outcome, like no infections, 1 infection in the house. You know, Fred gets infected, Fred and Giles get infected, you know, Fred, Giles and Sarah get infected, right? It's every possible outcome is given a probability by that vector. So you solve the whole thing in one go. And this matrix B, which I thought was nice to call the ball matrix, because Frank should probably have something named after him. And he didn't seem to mind too much that we sort of started doing that. It's got this structure. And I'll tell you what these things here are in the next few slides, but basically it has a block diagonal structure, so it makes the equation 2 even faster to solve. So the 3 bits of this are, firstly, there's a QI, that's your probability of avoiding infection from outside. And I assume that that's given by this kind of Gumbel or double exponential distribution, where the force of infection acting on you from outside is some force of infection everyone experiences, plus something that depends on your characteristics, like your age and so on. And then this is the baseline probability of avoiding infection from outside, which I will report in tables. Then you also need to know the Laplace transform of the duration of infectivity. And the reason the Laplace transform comes up is because of this this equation here, that basically the probability that your threshold chi that we pick being bigger than that is you integrate over all the times that you could have been exposed to infection for, and what comes out, because chi is exponential 1, is this term, which is the thing of an exponential 1, and that turns out to be, you know, the term that gives you a Laplace transform or something, right? And then the lambda between person 1 and 2 is assumed to be some baseline lambda here, plus factors that depend on person 1 and person 2, right, or person I and person j, right? And this log n thing here, this is the form that was suggested by Simon Koshmez as how to include final size, right, in these equations. But he didn't write it like this as a regression type problem, right? So Simon, this kind of writing it like a regression thing and using this double exponential link was Tim Kinyamjui and my that was the thing we were kind of arguing for in that paper I cited earlier on, was just a way of solving this problem of how to not have this is how we avoid stratifying every household as a household type, yeah, as we include individuals via these kind of covariates xi, right, and then we have regression coefficients beta and gamma and things that go on those covariates, right? So I know there's loads of equations here, it's too fast, but I'm just trying to give a basic idea. It's like trying to match up the Addy et al. Equations with a kind of regression framework so we don't have too many household types, because otherwise we couldn't identify an effect for every household type. There has to be some kind of separation into effect sizes. Okay, so we can produce a likelihood from all of that. We solve the Ball equations. Actually fitting to like over 1 million observations is difficult, so you want to solve that matrix equation quickly, and it wasn't possible to do MCMC, so we did approximate Bayesian inference using the Laplace approximation, which is again straight out of the 1980s. So, you know, here we are, and optimization methods that are also quite old-fashioned. So I'm sure there's the next step would be to try and get this onto some kind of secure cloud where we can actually do something a bit more modern. But at the time, the secure environments were just we're not cloud-based. So the UK government is buying a cloud, allegedly, or buying its own cloud so we can do more serious work. And then these are the estimates that come out of different effects. So the effects of external infection on people of primary school age, and external infection on people of secondary school age, and probabilities of infecting within the household as a function of size, and probabilities of avoiding infection. So I visualized those to make them easier to see. So it says probability of external infection in tranche 1, which is when our first lockdown was biting, was very low. Then we had this enormous second wave, and then we got vaccines, and so these low probabilities are kind of post-vaccine, and continuing lockdown from the second wave.
Thomas, you may have just said this and I didn't catch it, but when you say probability of external infection, is that during a specified duration of time?
Exactly, yeah. So it's a cumulative probability of infection.
Over
and
what?
You can also, and so it's those 6 tranches.
Right, how long each one?
They're variable because it was determined so that we could interpret time, to interpret time as an instrumental variable.
I see. So 1% in one tranche may not be the same daily risk.
So I've got it exactly. So I did that because I produced something better, but people said it was 10. So this is the better figure. That's more scientific, right? So I'll jump ahead and do this, but yeah, the kind of policy people struggled too much with this sort of thing at first, but now they're happy with this. So this is a bit like what it looked like, right? That we had the first wave that happened before this, then you have the second wave, and then these are the 5 and 6 that I was showing because they're in the peer-reviewed paper. But then we released all the measures and got Omicron, so there's still a lot of infection.
Have you visualized just the reported epi curve on top of that?
Yeah, so that's released by the ONS, so you can just download it if you want to play with it. They're point estimates of the prevalence over time, which is much less blocky than that. So this is just for the household analysis, but they produce a smooth curve of unbiased.
But does the household analysis sort of match the reported.
Oh yeah, yeah.
Uh-huh.
Pretty much, yeah. I mean, it's so coarse, and they're doing it every few weeks with
Yeah.
a really nice kind of smoother. But what you do see is that I think for some reason that I think we still don't understand, transmission was lower during lockdown. That lockdown, I do believe in a kind of seasonal effect. Personally, I don't think it's huge, but I do think in summer the virus dries out or something, because we didn't have any vaccines here, but people were still in their in transport. But it was summer, and it was quite a hot summer. So I do sort of believe in seasonality, not as the main driver, but I think we don't still really understand what weather it does for viruses. So I don't know. Then we hit the winter, and there's lots of transmission pre-vaccine, and then you see that some of this isn't just the MPIs; it's the vaccines are driving down transmission overall within households. That's consistent with the secondary attack rate analysis.
I'm sorry, I can't see what are the labels on the x-axis in the bottom?
Oh, yeah, that's cut off. It's P. This one here is P2. That's the probability of transmitting in a 2-person household.
Okay, thanks.
Probability of transmitting in a 3-person household, 4, 5, 6.
So can we interpret that is the y-axis like the secondary attack rate, or is this per person?
No, it's what Christoph Fraser called the susceptible infective transmission probability, because this is ignoring tertiary and quaternary and other higher order infections. So it's taking those out and saying if you just have 2 people, if you make everyone else, if you remove everyone else from the household, if you made everyone else in the household non-susceptible, and you just have a susceptible infectious pair, and in this case they're the reference class, which is 2 adults not working and patient facing and with wild type strain, how likely are they to infect each other?
Okay.
How likely is the infected to infect just the susceptible? So it should be more generalizable, because it takes out a lot of those things that can vary between populations. So when people try to meta-analyze secondary attack rates, that the meta-analysis tends to be inconsistent because people don't, because populations are different, right? So Christoph kind of advocated for this, like use the probability, because it should be more constant across populations. Okay, so then these are the effect sizes. The first thing is this trans other, is these are people where they had gene failures on the PCR other than the S gene. And so if your OR or your N fails, then you're probably really low viral load. So these people are not very infectious, right? So this is good. This makes sense. Then the relative transmissibility of S gene failure is low before the emergence of alpha, then high with alpha, and then when delta dominates, it goes down again, right? Because delta was more than alpha. Then we don't see that transmissibility in children is much different. And I think that's because it's mediated via viral load, right? I do think children are a bit less susceptible and infectious, but because we've kind of captured viral load in this term here, right, that's my interpretation. Other people have done pretty good studies where they show, so it's one of these like causation things you have to be careful about causes. Then what we do see is the susceptibility of the under 12s does go up when they're not at all vaccinated, because the UK vaccinated very late the youngest age group, which was kind of controversial, but I could kind of see they couldn't really, there's no easy answers like a lot of these things, but for whatever reason, empirically that didn't mean that there were more transmissions in that group, because they were, at this point, everyone else was vaccinated. And then we see the patient facing is really high. Like I said, they were very great risk of bringing it into the household, dropping because they were one of the first groups to get vaccinated. And basically the external risk for children then follows whether their schools are open or not. So it's this thing where this is quite a small effect when you look. So the ONS also did an analysis of how many contacts the children have. If you turn this into a per contact risk, it's very small, which is another reason I think children are a bit less susceptible. But because when they go to school, they have like 20, 30 contacts, it's quite a big effect, right? So this is a logarithmic axis on the bottom. So like I said, there's this kind of this wicked problem that we all want children to be educated, but there wasn't the kind of how was that? Is that moving around for you? Right, is that better? Okay, anyway, so yeah, there's some effect of schools, but it wasn't, some have somewhat diminished compared to adults, but not not complete. And then this is like saying, you know, because we've been doing this, this is just saying how an example of how the analysis has got updated for the latest tranche. Something's gone wrong with my zooming this, as in zooming the PDF reader. Anyway, so this is how the external thing looks over the whole epidemic. The transmission within households, it's like the vaccine fighting against the variants, you know, is the way I see this, that like, yes, the vaccines work, we kept boosting, but the variants keep escaping and keeping the transmission up. And like I say, we're at these absolutely enormous rates of external infection with Omicron. And then this is turning that the variable length of time into a force of infection, sort of, you know, what would be the implicit constant force of infection. Although the model doesn't assume a constant force of infection, it assumes a cumulative force. It gives you a cumulative force of infection. So you could draw any curve you wanted here, provided it had the area of the rectangle underneath the thing in it. And these are the effect sizes. So you also see, and unfortunately, the effect of Omicron and waning of vaccine coverage on the patient facing. So we've still got a problem with transmission within households with Omicron, within hospitals, sorry, with Omicron. The hospitals are in a real state. And yeah, another S gene change, because Omicron was S negative, then the new Omicron is, which the next tranche of this should be about, is S positive again. So we've had this flipping, and the schools are continuing to be a kind of significant, now really quite big locus of transmission compared to the rest of society. So thanks to the ONS team, who I think have been wonderful. We've got a little group now in Manchester. Anyone fancies visiting, if you like soccer, football, and rain and indie music, Manchester's very good for those things. It's the Seattle of England, roughly speaking. So sort of rainy, but quite culturally, and quite sort of, you know, lots of nice vegetarian cafes and things as well. Right. It's just tech industry, but not quite as big as Seattle, sort of thing. Yes, this Juniper Consortium was wonderful. The Royal Society were very flexible about the fact that I'd said I was going to go and work with IBM and learn how to learn a bit of machine learning, and instead I just kind of got some, you know, IBM were very, very nice about giving me compute time and expertise, but it wasn't I had this kind of fun. My plan had been to go and just hang out at their campus just outside Manchester and kind of learn some cool machine learning. And in the end, I just ended up doing stuff that I was doing anyway and didn't learn as much from them as I'd hoped. But they were very, I should add them here, they were very generous with compute time and support for some of the software engineering. Alan Turing Institute, yeah, UKRI, these other people who helped, Sage. And it's been nice. SPI-M is this modelling subgroup of Sage. That I think has been quite a good thing. And yeah, thanks, Lauren. Hope you have a nice rest of the day, and thanks everyone for the invite and your time.
Thank you so much. So I think we're a bit over time, so feel free to drop off if you have to, everyone. But I'll stay to kind of coordinate any questions. Does anyone have any kind of pressing questions? And maybe we could just take like 4 or 5 minutes more. So I had one question, which was, I think I missed how the kind of sampling worked. So originally I thought there were like regular intervals that you were actually testing within households, but I don't think that's actually what was happening. Were you just tracking?
So okay. They're tested weekly. They're tested weekly for the first month, then approximately monthly for the rest of the year. But the data can be linked with any other testing they've done themselves. And because we have this mass testing, there is an issue with ascertainment. We probably miss some infections. And that would be a nice thing to try and get MCMC working for, so we could impute missed events. But there's, you know, at some level, you have to just take the data on face value and say, well, if we assume we've ascertained, this is assuming we've ascertained all the infections. But in practice, it doesn't tend to change the relative conclusions that you draw. So the thing of the relative effect size is actually relatively constant, but the overall secondary attack rates can be quite different between study designs. So there were two other study designs, big household studies done. One was called hosted, which was passive linkage in the test and trace, and that tended to find, the equivalent probabilities that I was showing were about half of ours, because it was just a passive surveillance relying on people reporting their household members. Then there was an active study called attack, where they sent people daily into households, which it turns out is what Denmark did this, the Danish household studies, have you seen them? They did daily testing. And that generates slightly higher probabilities than our one. So we think our ascertainment is somewhere between, as you'd expect, sending people in daily and completely relying on people to just report their household members to the system themselves. So, I think we can take the attack and the Danish ones as pretty much gold standard, that we're slightly underestimating total within household transmissibility. But it wasn't like night and day. It was it was slightly smaller and slightly bigger. What's interesting is a lot of people are fatalistic, and I think once it's got into your household, you've all got it. But actually, attack rates always tend to be below 50%. And I think it's what Hope Simpson found, that even siblings didn't always infect each other. The stuff that was very infectious, like measles, it's slightly surprising. And I think we still don't understand something about the physics of how the germs get out, and how random, stochastic, whatever that is.
And did you have any estimates of like caregiving status, or like, do you differentiate between like parents to children versus children to parents, and I guess like siblings?
No, but this is, I think, very important. And the other thing that the ONS does is it holds quite detailed data on like structural, what the structure of the house is. So there's also like, are people overcrowded? That we should know that. And because all this was done in a rush, and we all had lots of other things we were doing for the pandemic, it was tricky. But the looking at actual relationships, and there's this whole thing is fresh air, the new sort of clean water, right? That like Jon Snow's big thing was, we should drink clean water. And there's a big thing about are people, could we really knock down the rates of respiratory transmission by being in better ventilated spaces? And I think there is probably a lot of like, maybe we should be trying to retrofit houses with better, and so looking for some really sound, if there was some really sound evidence that the houses that you would expect to be more crowded and not have as much fresh air passing through them, have more infection, that was a big thing we wanted to look at, and still can, right? We're hoping to do that sort of thing, but it wasn't really. A lot of things were geared towards policies that might change, whereas the government wasn't going to start retrofitting houses during the pandemic. But I think there is a bit of talk of, should we just be thinking about better ventilated? I don't know whether that's happening in the States, but like I say, Kath and people with the thinking about these things being airborne and airborne mitigations, could we all have a bit less flu? Could we all have a bit less flu and feel less woozy? Like these online seminars are in a way good, because you used to kind of crowd into the seminar room, and I'd always start feeling sleepy, and that's because if you had to put a CO2 monitor there, CO2 just shoots up, right? And I don't think it's healthy for us to be in these spaces that are poorly ventilated. So it could be one of these double wins where we all get less journey and feel more alert if there's more air passing through.
So the US, I think, is having some conversations about it, but I don't think it's gaining much traction other than potentially in schools and facilities that are like that. One interesting thought is that I was thinking is the seasonal dynamics. It could be climactic overall, or it could just be that people were having their windows open in summer versus not, which would, as you say, increase ventilation. And so, that would explain those dynamics, right? Like there was fewer.
Yeah. So that's the issue, is what's the causal diagram? And for flu, there was this huge argument about, like, it is true that viruses don't like drying. I mean, like, viruses don't like UV light and fast evaporation of the water that they sit in. But it's how big an effect is that versus the impact of the hot weather on human behavior is quite hard to disentangle. We were talking to meteorologists about that. And there were some interesting ones where you can look at, because I said, we had frost, the UK kind of experiences these really odd flips where you'll have like a week in the middle of otherwise quite hot weather where it's sort of icy or snows or something. So we're actually hoping there might have been a few natural experiments that happened with those kind of odd bits of weather that are kind of, might help you see.
Mm-hmm. That's cool.
But yeah, it's hard because I just think there's a lot we don't understand, right? Because you guys have probably been thinking for the last 2 years as well. Right? It'd be nice to know more about all this stuff.
Yeah, we understand a lot about COVID, but the more I go through the pandemic, the more I think we don't understand anything. But anyway.